What'd you guys think of that case study yesterday? I know I did get one private message like, do we really have to know all this stuff? It all seemed like there was a lot that seemed unfamiliar. Yeah, you guys, like from here on out, it's kind of a new territory for you guys. Prior to second semester, most of that was like a year of biology. What you've already done, there wasn't a lot of new. It was just faster. We added a bit more detail here and there, but I would say from here on, you're going to get a lot of new. So some of it, like DNA structure, is going to be familiar, which is why I fly right past it and don't really spend time on it. And I'll slow down and I'll spend more time on the gene regulation. That's going to seem more unfamiliar to you. So second semester is going to require a little bit more effort, maybe, in the understanding part, because it's not so much review. And I think first semester, a lot of it was review. Okay. So glad you guys enjoyed that case study. I really like those case studies, and it's cool because it's like real world things. There's a lot for biology, not as many for the AP world, but that one I thought was a good one for us because that's what we're getting ready to get into. So I want you guys, you have an activity that looks like this. Actually, I'll go through the agenda, then we'll do that. We're going to do that activity just to get us a chance to talk about the structure of DNA in a discussion format rather than in a lecture format. And then over the weekend, I want you to read and update your notebook 12.3 and 12.4. So I've taken your notebook here, and if you scroll to slide 12, I believe, here, if you could just grab hold, select one, scroll it all the way down to 18. Control Shift allows you to pick it all at once. You can do them all individually if you want. I right click, I copy slides, and then I'm going to open up the one that you already own. Okay, so you'll go into your drive and you'll open up the notebook that you started with, you know, the picture and the description of each, and then right click. So here I am at slide 11 for you guys. I'm going to right click and I'm going to say paste, or you can do Control C, Control V, whichever. So now I have those slides added in. I can't make I could have made a copy for each of you guys, but I wanted it just to be in one notebook for you, for when you're studying, you don't want to look in a bunch of files. So you can just paste that into your old one. Now I'm going to go Control Z, and that's Edit Undo. And then when you guys turn that in, you will just insert that into today's assignment. So chapter 12 notebook update. Notice here's your original, the set I gave you, but it won't show up as a copy for you. So when you turn it in, you have to make it an attachment, and you can reattach that, okay? We're going to be doing transcription is in your, I'm looking in the chat box here. Transcription is going to be part of today's or this weekend's reading. And that is going to be next week's discussion. So I didn't really know what would be best. If you guys have a different format, that's fine. One, we're doing RNA structure. I want you to be able to compare and contrast the structure and function of RNA and DNA. So some of it's going to be similar, some of it's different. A Venn diagram would work. And I know you guys might do your own format and just paste that on there. That's fine if you don't like my format. Types of RNA. I had scanned this book because I came to help my mom this week, and I scanned the pages when I was at home, but now I can't find the scans on my phone. So I don't know exactly what is in your book. I know it'll have mRNA, RNA, tRNA. I'm hoping it has some others like snRNA. So if your book includes any others, you can talk about those there. And then transcription, remember, we want to be able to understand the entire process and like you really do need to know step by step, because if you're given on the AP test, they like to throw a wrench into the system, right? They're going to say some gene is messed up that affects an enzyme somewhere along the line. So you have to know where everything is in that process so that you can say if part C is messed up, then D on is not going to happen, right? And A through C might back up. Make sense? So we really do need to know all of the things that are happening. So I said, like, include a description of the process. Most of us did really thorough descriptions. I thought of the scientific methods in this chapter. Some of us maybe were missing key points that I think will be important when answering questions in the future. So make sure you're trying to describe this process as though you're explaining it to somebody else. Okay. Key terms should be included, include an image. Transcription, translation, mRNA. There should be a central dogma. That's the overall flow of information. You might not fill an entire page. I get that. mRNA editing, that might be, this is going to be one where some of it you know, and then some of it is going to be additional information for you. So again, just like last time, I like seeing your handwritten notes too. So if you want to just copy the, scan your note and put it on there as a pic, that's fine too. I like it. Okay. Any questions on that? Can anybody tell me somebody tell me about Griffith's experiment? I wasn't going to talk about Griffith's experiment today, but I wanted to talk about Avery, Mclaughlin, and Mccarty, and I didn't feel like I could talk about them without Griffith. Can anybody tell me about Griffith's experiment? What was he trying to do?
Create vaccine for pneumonia.
Right. So a lot of people are going to, since it's in this chapter of DNA, a lot of people automatically think it has to do with DNA, but he wasn't even investigating. I guess I'm going to go out of order here. He wasn't even investigating DNA in the first place. He was trying to find a vaccine to fight pneumonia. And instead, he found this transforming factor, which he didn't even know was going to be of importance yet, right? He was kind of stumped at the end of his experiment. He never really finished. He set his research on a shelf. World War II happened. His lab was bombed. He never finished. So for us, he just kind of ended with this big fat question mark, which is what a lot of science experiments do, right? And then you start a new experiment based on that question mark. Can you tell me about the S and the R strains? Anybody? See, this would have been a good Nearpod, and because then you guys all have to answer, but let's do it like we're in class and I'm asking you questions and you're raising your hands and you're answering. Tell me something about, you can chat it if you're afraid, S and R strain. We're going to be going back, talking to each other here in a couple weeks. Which one was, what does S stand for?
Smooth.
Okay, so you had a smooth and rough, thank you. Which one was harmless? Harmless.
The rough.
Good. Okay, why was the smooth smooth? We have to understand this to understand the process.
It had a capsule around it.
Yes, it had a capsule around. So the capsule, the smooth was deadly, virulent. Why was the smooth deadly, do you think? Any ideas why that smooth one would be deadly and the rough was not? Has to do with the capsule, but it's not like the capsule was this virulent factor that was causing us to be sick. The capsule was a protection for that bacteria. So it protected the bacteria against our own immune system. You guys, I'm probably going to be going long today because we're just going to talk. So that capsule protected it against our immune system. And so we didn't break that bacteria down. So it was able to cause disease. Where the rough didn't have that outer coat, it wasn't protected. So our bodies were able to attack it and break it down. Okay, so hopefully you guys recognized that he had the S strain and it killed, the R strain and it didn't. Then he heat killed, because he's trying to make, remember, a vaccine is like a dead or weakened version of the virus, right? So he heat killed it so that the DNA, or he didn't even know about DNA, we can't say that. He heat killed it and then was hoping this weaker version, then he would be able to use it as a vaccine. So he put that into the mouse and the mouse didn't die. So the once deadly version was no longer deadly. But when he combined it with the rough strain, which was not deadly to begin with, it caused the mouse to die. Now, he didn't understand why that was happening. He didn't know then that the rough strain was changing its entire molecular structure, right? He just knew that this once non-deadly version is now deadly, and this is a transformation. One of our key labs, this is why I decided to go over this today. One of our key labs is a bacterial transformation. So we have to understand that. It's a change in phenotype, right? So this, what we know now, the R strain grew a capsule. So here's the R strain that was not deadly to begin with. When they were combined, it grew a capsule just like the S strain, which allowed it to be protected from our immune system, which allowed it to cause disease. Okay, so that was the transformation. So he's like, why did this happen? What caused the transformation? I don't know. My lab is bombed. I can't keep going. I give up. Okay, so we know now, right? We know about that transformation factor. We know that it's DNA. Back then, back in the 20s, they did not know. And they assumed that genetic codes were either carried in nucleic acids, this material that they found in the nucleus, or it was in the protein coat, in the proteins of animals. Majority of scientists thought it was proteins because proteins are involved in so many processes, like all our enzymes, all our functions, right? So majority of scientists at the time thought proteins were the transforming factor. So that's the two ideas. Was it protein or was it DNA? Now, for our lab, we know that it's DNA already. And what's happening is that this bacterial cell is rupturing. When he heat killed his Streptococcus pneumonia, it ruptured the protein coat. DNA was denatured because of the heat, and it broke apart. The bacteria are actually permeable to DNA. They can pick up DNA from their environment. They can pick it up from the tables. So that's one of the reasons we got to bleach our tables, because that DNA can be absorbed right through the bacterial cell walls. So it's absorbed by the bacteria, and then it's incorporated into its own genetic genome, right? Its own chromosome. Once it's in its chromosome, it's going to be replicated, transcribed, and translated and make the proteins that it codes for. Okay, so that's the transformation process. Okay, so here's where Avery, Mclaughlin, and Mccarty come in. And I didn't have them on your notebook because I was thinking our book didn't cover them too much. And then I looked at your pretest and I'm like, oh shoot, I thought the question I had given you was a Hershey Chase question, and it was an Avery, Mclaughlin, and Mccarty. So I felt like I had to talk about that today, because after today, we're moving on to other stuff. So Avery, Mclennan, and Mccarty, what they did, they were really interested in Frederick Griffith's work. They're like, was it DNA? Was it proteins? Was it a carbohydrate? I don't know. What caused the transformation? So what they did was they isolated each of these molecules. So they used an enzyme that would destroy. A proteinase is going to destroy the proteins. Ribose, that's going to destroy RNA. Deoxyribose, let's say DNA nuclease is going to destroy the DNA, right? So if we get rid of the protein and transformation happens, was protein the transforming factor? If you got rid of the protein and it still happened, it wasn't the cause, right? RNA, used an enzyme that destroyed the RNA, so now RNA is gone. Transformation still occurred. Was RNA the transforming factor? No. DNA. Use a DNA nuclease, which we use today. We use these nucleases to isolate genes in the lab. But they use this to destroy the DNA. Transformation did not occur. So if there's no DNA, there was no transformation. Therefore, the DNA must have caused the transformation. Agree? Agree. Very good. Now, they weren't very convincing because the whole scientific community still was like, I think it's proteins. I don't know what is going on with your lab. So they were the first to identify DNA was the genetic material, but it was not accepted at the time until Hershey and Chase's famous isotope experiment. And I'm not going that far today. I just wanted to get this far so that we could look at that question that was on your pretest. Okay, so this just summarizes that same thing. We got rid of the protein, no transformation. Therefore, protein didn't cause the transformation. Or transformation still occurred. So it wasn't the transforming factor. And I just explained that, so you don't need me to say it again. So now let's look at our question. In the 1940s, Avery, Mclaughlin, and Mccarty transformed non-encapsulated, i.e. rough, bacteria into encapsulated. I have to talk to the text when I interpret these long paragraphs. You should do the same. So transform non-encapsulated bacteria into encapsulated forms, i.e. the S strain, by growing the non-encapsulated cells in a culture containing the extract made from the dead encapsulated cell. Sounds like Griffith's experiment, right? He killed the S, put it in with the R, and it transformed to the S. So he used 3 different experiments. In the first, blah-de-da-blah-de-da-blah, he used, this is basically set up just like the original, right? Dead encapsulated with the non-encapsulated, and both in the jar, he got non-encapsulated. It would be on a petri dish, non-encapsulated and encapsulated. So there doesn't seem to be, there seems to be some of each, right? Then he's going to use that enzyme that kills it, like the Avery Mclaughlin. This is Avery Mclaughlin. So he takes those dead encapsulated ones that originally caused transformation. He used the protein degrading enzyme, non-encapsulated bacteria added, both still showed, like the original form. That means transformation is occurring. Extract or product 3, this is the lab I just explained. He used the DNase and he got rid of the DNA. And then he got only non-encapsulated bacteria to grow. So there was no transformation in the third one. Okay, first of all, I want to ask you, what is the purpose of procedure 1? What's the purpose of procedure 1? Chat or shout. Is it an experimental setup? It is a control group. Bonus. Yes. So nothing had changed in experiment 1, procedure 1. That's our control group. That's the one we're using from comparison. Procedure 2 had the same result as procedure 1. So the independent variable made no change. What was the independent variable?
Protein degrading enzymes.
Yes. Okay. And then procedure 3, this time the independent variable was the DNA enzyme, and we saw transformation occur. When I compared it to the control, it was different, right? So that independent variable made an effect. Moving on. A reasonable conclusion to draw from this experiment is DNA replication is semi-conservative. DNA is a double helix. DNA is translated into protein. Mutation is a change in the genetic material. DNA is the genetic material. Which one basically answered the questions that was being asked?
E.
Yeah, 7 of us got that right. So we're off to a good start. DNA replication is semi-conservative had nothing to do with the lab. That didn't talk about how DNA replication occurred. It didn't talk about if it was all conserved, not conserved, yada, yada. Didn't talk about the structure of DNA, double helix. So we can right away throw out A and B as being totally irrelevant questions, right? DNA is translated into protein. That's maybe in the wheelhouse here. Mutation is a change. That might be in the wheelhouse. But if I looked at what were they trying to prove or what were they trying to find out, they were asking what is the transforming factor? So my conclusion has to answer that question. So even though you guys haven't read that part, right, you're going to get experiments that maybe you haven't read about. This is kind of how you have to think through those. What was the question being asked? Okay, so DNA is the genetic material was the answer. Okay. So I'm not going to go into Hershey and Chase, or yeah. You have my entire lecture of this. I put it in Google Classroom. That's historical studies. I want to jump to, what do you know about DNA's structure? I got to scroll up. So in the chat box, just throw some things at me. What do you know about DNA structure? Anything at all, or shout it out, because that can be easier, really. Does anybody know the shortcut key for your mute button? There's a shortcut. I forget it myself. So we know it's a double helix. Give me something else. C goes to G. Who said that? I mean, I know literally Cameron said it and Grace said A equals T, but what scientist gave us that information? Purines and pyrimidines, good. Oh, good, triple bonds, double bonds. Chargaff, yep. Okay, so purines, which one's smaller, purines or pyrimidines? Which one's the smaller molecule?
Isn't the pyramide smaller?
Yeah, it is. It's a double ring structure and it's a single ring structure. And that's one of the things that Watson and Crick used. If DNA maintains the same distance between the two, you couldn't have adenines, you couldn't have purines bonding with purines and have a big wide section, and then pyrimidines binding with pyrimidines and have a skinny section, right? The only way for it to maintain a constant width would be for a purine to bind with a pyrimidine. And then Chargaff found, he didn't say that the two bonded together. He simply found that they occurred in equal amounts. And because they occurred in equal amounts, Watson and Crick went the next step and said they must bond together. Okay. Okay. So let's see if we can do some. You guys have these 3 slides. This is the only thing you're turning in today. So take a moment, please. Let's see what we know. This is our review piece. See if you can do a click and drag and label this DNA molecule. I am going to share my screen with you. I'll show you why. I had forgot. I missed this when I made this click and drag share screen. Inside the molecules, so in the big box, you'll use your purine or pyrimidine. And then I had whited out inside of the molecule itself, a box, and that's where you would have put A, T, C, or G. So you can just grab one of these guys and put it on top of, if it was not that. Make this adenine, thymine, cytosine, or guanine. That's why there's 4 extra. I forgot about that when I made it last. You guys can make it A, T, C, and G instead of the whole word if you want to make it shorter. So I think you grabbed the phosphates and the, you grabbed the phosphate one real quick. That was pretty easy. This is a single ring, so it's the smaller molecule, so it's the pyrimidine. For me, I always thought the bigger word was the smaller molecule. Adenine, I don't want yet. Maybe I should have done numbering of carbon atoms before I actually do the 5' 3' end. The triple bond here, double bond here, okay? Those are hydrogen bonds. So we can grab that one right away. Now that helps us determine whether something is adenine, thymine, cytosine, or guanine. So if there's a triple bond between them, like this, that tells me that this is cytosine and guanine. So then I have to think guanine is the adenine, or guanine is the purine, so the bigger molecule, right? So that makes this guy guanine. The single molecule is the pyrimidine, and cytosine, the C's or the Y's, cytosine is the pyrimidine. So that helps me label that one. Here's a double bond. So that tells me adenine and thymine. The single ring is the smaller molecule, and therefore the pyrimidine. Thymine is the pyrimidine. And the double ring is our purine, and adenine is our purine. So you could match colors. You might have labeled these two or these two. That's fine. This is pointing at the double ring. Double rings are the larger molecules and therefore the purines. And oops, that's not what I thought I grabbed. I realize now my spaces are too big. So now we have deoxyribose, the sugar itself. Looks like this arrow is pointing at this sugar itself. It's upside down on this side. They're anti-parallel. And the phosphodiester bond exists where? In between nucleotides. So it would be this one here, between the phosphate of one and the 5th carbon of the, okay. So now you got to determine the 5' and the 3' end. Which one is on the left? What do you guys think?
The 3 end.
Yeah. And that's why I should have done the numbering of carbon atoms first. So that side was the 3', this side is the 5'. So when numbering the carbon atoms, you start with the carbon after the carbonyl group. So the carbonyl group is right here, the O. Our C double bond O lost a double bond when it became a ring. So I start numbering clockwise after this oxygen that is in the carbon ring. So I'm going to start clockwise, meaning this is going to be my first, and you say prime. That one with the parentheses, that's your 1' carbon. So that's how you refer to them. Going clockwise, we have our 2' carbon, 3, 4, and where you have organic structures, where 2 lines come together, there is a carbon. They don't write the carbons in because organic is the study of carbon. So they're assuming everywhere there's 2 lines, unless they draw in a different letter, where 2 lines meet is an assumed carbon, right? So this is going to be 1, 2, 3, 4, and 5 right here. So we'll do this one together, and then the next one you guys are going to do on your own. Now, the reason why we have to number these is so we can understand orientation of DNA. And now that you have a way to reference each of these carbon atoms, I can say, I can say the carbonyl is between the 5' and the 1' carbon. I can say there's a hydroxyl on the 1', 3', and 5' carbons. Okay, I can talk about this molecule more specifically now that I have a way to reference those carbons. So specifically, what is the difference between these 2 molecules?
I said there's a hydroxyl group on the 2' on the ribose molecule.
Okay, there's a hydroxyl group on the 2' carbon of ribose versus deoxyribose. So is it missing a hydroxyl group entirely, or is it only missing part of that hydroxyl group?
Doesn't deoxy mean without oxygen, so there's no oxygen?
Right, right. So you could say there's a hydrogen instead of a hydroxyl, or you could say that it's missing an oxygen on the 2' carbon. Yep. So that's where the deoxy comes from, is losing this oxygen in particular. So the difference between DNA and RNA, one of the differences is the sugar that's used, and that's the whole difference between those 2 molecules is a single oxygen. So now that we've labeled this carbon, this sugar molecule, you take a moment and label the 3' and the 5' ends of this section of nucleic acid. And I'm going to scroll through you guys to see timing wise where I'm at. Can also kind of see if you guys are on the right track. So I think we all got where the 3' carbon is, or oops, that's a 5 that I grabbed. So let's move that up here. It's at that connector, right? And the 3', maybe I made my letters a little big, I don't know. 3' is there, and I could keep going. I'm going to bring this one down here. So at the hydroxyl is the 3', the phosphate is the 5'. Which carbon atom is the nitrogen molecule attached to? The nitrogen molecule. The nitrogenous base. Adenine, thymine, cytosine, guanine. Which carbon atom is it attached to? Commonly missed question. Nitrogenous base. Is it 1', 2', 3', 4', 5'? Yes, it is. So now we are better able to talk about this molecule, right? We can say that the nitrogenous base is on the 1' carbon. The hydroxyl is on the 3' carbon. The phosphate is on the 5' carbon. Where is the phosphodiester bond? It is between the 3' carbon, right? It's between the hydroxyl of the 3' carbon and the phosphate. So if you remember all the way back to chapter 3, we talked about glycosidic linkage with carbohydrates, peptide bonds with proteins, ester bonds with lipids, and now we have phosphodiester bonds between the phosphate, that's the phospho part, and the hydroxyl of the 3' carbon. So this gives us the directionality. We talk about DNA growing in the 3' direction. Add free to the 3, right? So that means this side of the molecule is growing in the downward direction. Anti-parallel, DNA is 2 different, right? So anti-parallel, the other side would be going in the opposite direction. 5' to 3'. It's red 5 to 3. It's growing. I'm sorry, it's red 3 to 5 and it's growing 5 to 3. That gets confusing. So we did this labeling, and I just wanted to show you the directionality. So I'm opening this PowerPoint and I'll just hit like the slide. So you can see the directionality here, right? So 5' is up on this side, 5' is down on this side. 5' has the phosphate. Complementary because the adenine bonds with the thymine, and that's the only way they could go. They're parallel, but they're anti-parallel. One is upside down compared to the other. That anti-parallelism is what ensures you will always make identical copies each time, okay? So that's what I have to say about that. This other piece, I believe this is all review. So I put the PowerPoint in your Google World as a reference in case you forget. Whew. How are we doing so far? That was a lot of information, right? Anybody need to take like a 5-minute break? You want to keep going? Plug. Okay. All right. If you need to pop up and go to the bathroom or anything like that, feel free. I understand if you want to get up and stretch. Oh, stretch. Okay, here we go. Okay. So semi-conservative, you probably read about, off the top of my head today, I cannot think of his name. It's not Sturtevant, but it is an S name. Anyways, he did a study with, again, radioactive isotopes, so he could follow the molecules, right? So he made one set of nucleotides heavy, and he was able to follow where those new nucleotides that were being added in were. So he could see there was a 50% heavy isotope and a 50% of the regular isotope in the first generation. And so that led him to the whole semi-conservative concept, in that one of the DNA strands is conserved and one is replicated. That's something you guys already know. Something we maybe have not talked about yet is versus bacteria, right? So in eukaryotes, we have multiple points of origin. So when we're replicating our DNA, it doesn't just start at one point and then become a zipper to the other point. It starts in many points because we have a lot of genes to cover, and it would take a long time if you started at one and had to go the other. So it's going to divide and conquer basically. So you can see these replication bubbles. So you can refer to point of origin or origin of replication at the beginning of the bubble, and then it's going to work its way out. If this was a bacterial cell, it only has one point of origin, and it goes around the circle. So it replicates the chromosome all at once. So that's one of the differences. You know helicase, you know polymerase, you know ligase. Those are old news. New ones for you should be maybe or maybe not primase. The fact that there's multiple polymerases and telomerase. Those are new enzymes for you compared to first time around. So we'll talk about what each of these do. Remember, the name itself tells you something about the molecule. So even if you've never seen it before, come test day, anything that's an ASE ending is an enzyme. The first part tells you whatever it does, whatever it acts upon. So the helicase acts on the helix, right? So it's going to unzip the helix. Primase is a primer. So we'll get to that. Polymerase tells me that it makes a polymer. If it's DNA polymerase, it makes DNA. If it's an RNase polymerase, it makes RNA. Ligase seals the gap, and telomerase is going to be found at the ends, and it conserves the telomeres. So here's where we're at. Polymerase 3 is your main builder. So it's the one that when in freshman year, when we just talk about polymerase, this is the one we've talked about. So it kind of feeds DNA through. It's kind of like a hand, and it adds, part of this enzyme is grabbing nucleotides and adding it to the growing DNA strand. So that goes pretty quick, which means it's likely to make a number of errors, right? If you go quickly typing, don't you make a lot of errors? And then you got to go back and edit it. So polymerase 1, and this is just like numbers because we found them at different times, right? So polymerase 1, notice it's much slower. This is the one that's going to make those edits. So I had a typo and I have to go back and fix it. So that's polymerase 1. Edit, repair, and remove the RNA primer, which is kind of like a placeholder. So this is an example. Thymine dimer, it accidentally grabbed 2 thymines, right? And you only wanted the 1. So polymerase 3 adds the nucleotides. Polymerase 1 comes and says, whoa, that's not right. And it takes it out, takes out the damaged part, and it puts in the correct piece. Ligase seals the gap. So this makes it so that we have much less error in our DNA replication. So far, so good. Okay, so E. coli, which is a bacteria that we use a lot in the lab, it replicates every 40 minutes, which is why we use it in the lab, because we can make lots and lots of genes. And if we can make lots and lots of genes, we can make lots and lots of proteins. So it's very quick. They have 5 million base pairs in a single chromosome, and the eukaryotes have much more, 4 billion bases to get through. So it's remarkably accurate due to that polymerase 2. So there are error rates, and you guys already looked at some error rates in our case study. You saw some mutation. So here's one of the new pieces for you, the single-stranded binding proteins. You already know about helicase. You know that it unzips, it breaks the hydrogen bonds. We've seen that. We probably didn't talk about single-stranded binding proteins. They're here. They're holding the DNA open so that the new nucleotides can be added in this bubble. Okay, so this is our bubble of replication. So helicase opens it up. The yellow is opening it up. These blue circles, these are your binding proteins, and they're holding that open so that new nucleotides can be added on the inside so it doesn't seal it up. So this is that directionality, right? We're adding free to the 3 new nucleotides, free floating nucleotides are being added to the 3' carbon. The 5' phosphate is being added to the 3' carbon, right? So it's red. This is the dark blue is my reading strand, right? It's reading 3 to 5, but it's building 5 to 3. So you have to be really careful with that. Remember, they're opposite. Here's your single-stranded binding proteins. The helicase is breaking it open. So this leaves you a leading and lagging. The fact that they're anti-parallel, right? My 3 to 5 direction is opposite on each side. So one, oops, I took a picture rather than hanging it up. One will follow the replication fork. So this one is able to add nucleotides continuously. So this is called your leading strand. The other side, because it's going in the opposite direction of opening, it ends up in fragments because like I unzip and only this much is available to add nucleotides. Meanwhile, it continues unzipping above here. So now I have to backtrack, get that piece I missed. Meanwhile, it continues to unzip. So that side has to keep going back and filling the gaps. So this is the lagging strand. So it creates Okazaki fragments due to the scientists who discovered the fragments. These pieces here are your Okazaki fragments. Before we start replicating this, we lay down an RNA primer. This little red piece is the primer. This gives, because remember you need that 3' carbon to add the phosphate to, right? And I don't have that over here. So it, this gives it something to bind to. So you put down an RNA primer, which later will be removed. Ligase will remove that primer and bind, like fill in the gap. So this is showing you 5 to 3' is leading, 5 to 3', but it like at one, it builds here and then it's got to come back and fill in what's missed. And then it's got to come back and fill in what's missed. So that's the lagging component. You put down an RNA primer to give it an anchor, basically. So I talked about that already. This is all about the directionality. So to save time, I feel like I already talked. I like this image. This is the famous polymerase 3, and you can see these ends here. So these are the finger-like portions of the hand. So it has a circle portion. The DNA is running through here, right? DNA is going through this part, and then these are grabbing new nucleotides and adding them to the growing strand. So that structure function, right? So we've heard of ATP, adenine triphosphate, right? Each of your nucleotides has its own triphosphate form. So each reaction is actually powering its own reaction. So when you add a cytosine, it has a cytosine triphosphate. When it bonds, it breaks its own phosphates off to release the cytosine nucleotide. It's kind of cool. If you launch this in PowerPoint, it's animated and it goes one at a time. I didn't launch it in PowerPoint. So you would see how each of these has its own triphosphate, and then you would see the double phosphate break off and leave behind the single phosphate that's part of that nucleotide. That's a mess if you don't do it. Okay. Leading and lagging strand, you refer to them as continuous and discontinuous. So the leading strand is following the replication fork. It's continuous because it doesn't have to stop along the way, right? The lagging strand is discontinuous. It's broken apart. I'm going to show you an animation. Oh, wait. Well, I'm going to delete that because I put it here and I already cut it. So this is your RNA polymerase. or your DNA polymerase, sorry. These are those hands I was talking about, works like fingers to pull things in. Notice the DNA going in. Notice the continuous strand coming out. Notice the discontinuous strand. I like this animation because you can see how it has to go back and backtrack. Somebody asked me at the beginning of the year in that, like, that you guys had to ask me questions. They said, what's your favorite unit to talk about? You got it right here, kids. So anyways, this piece, you'll see it's coming out and only this much is available to replicate. So it replicates that. Meanwhile, you'll notice it continues to unwind behind it. And that's why it has to backtrack. So I like this animation because you can see the backtrack.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells.
Leading,
You are looking at an
lagging.
assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called Helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules. Using computer animation.
Okay. So that was, I think that's super cool that you could see, I think it just gives you a better idea of that lagging strand. So this is the DNA. It was coming in from the left, right? This picture is opposite though. Helicase, the leading strand is following that helicase. It's building 5 to 3. It's reading 3 to 5 in order to see the 5. In this case, only pieces of it are available at a time. So it has to replicate in pieces, right? So we saw that. Which leads us to another one of your pre-test questions. When DNA replicates, each strand of the original DNA molecule is used as template, blah, blah, blah. Which of the following figures most accurately illustrates enzyme-mediated synthesis of new DNA at a replication fork? So when I look at this, I have to remember DNA is anti-parallel. I have to pay attention to the direction, the 5' and 3'. And I have to remember that one is going to be leading and one is going to be lagging. So right away, I can eliminate 2 choices. Which 2 can I eliminate right away?
A and C.
Correct. So now we look at B and D. Which one do you want and why?
Wouldn't it be B? Because I don't think it works backwards. Like D does.
The direction of the new replication. In this case, they're both going toward the replication fork. That means this one is building 3 to 5. And this one is building 5 to 3. We know they all have to build 5 to 3. Knowing that they're anti-parallel, that should eliminate the choice B. In D, the leading strand at the bottom is going toward the replication fork. And the lagging strand at the top, because it's in pieces, I know that's the lagging strand, is at the top. And it's going away from the replication fork. Does everybody see why D is the correct answer when you look at directionality? Okay, very good. All right then. Oops. I guess there is a little bit more, sorry. So one of the enzymes, we have the telomerase. So we still have to talk about telomerase. So we're using that primer that we have to get rid of, right? So here, because there's no 3' end to continue, you're going to constantly lose a small portion of your DNA each time it's replicated, right? So our DNA actually has a lifespan, and this is how we get into aging, that whole aging process, wrinkles, tissue failure, because you're losing a small section of that growing strand with each replication. Okay, so because we can't replace that RNA primer at the very end, this is called chromosome erosion. Loss of bases at the 5' end, it's going to get shorter each time, which means it can only replicate so many times before it starts getting into coding. Like this portion is just all added content. It's not coding for any particular proteins. But after years of replication, you're going to start getting into coding genes. And that's where we start to have the aging process kick in. Okay, so the telomeres are those pieces at the end. They're actually there to like protect the DNA from damage of enzymes within the cell itself, as well as to give it a lifespan. So the repeated units of non-coding sequences, so it's kind of like junk DNA, you might consider it. Telomerase, ASE ending, creates those telomeres. It extends the length of the DNA. It extends the age of the DNA. Stem cells have a lot of telomerase. Cancer cells have a lot of telomerase. That's what gives them an extended lifespan, because they can continue to create telomeres at the end of the DNA. So they're adding DNA at the 5' end. Different cells have different levels of telomerase. Okay, another pre-test question you had. Enzyme used to position nucleotides during DNA replication. Which one is it? Which one's going to add the new nucleotides?
Would that be B?
Yes. So we know it's a polymerase. So right away, I'm limited to choices B and C. The first part tells me what kind of nucleotides it's adding. We use RNA polymerase during replication, this week's reading. DNA polymerase for replicating DNA. DNA ligase, what does it do?
It glues together the fragments.
Yes, very good. Okay, restriction enzymes, we won't get into those until we get into DNA engineering. But what they do, they restrict DNA to a specific gene. So we're going to get into that later. When we want to study a certain gene, or we want to make insulin, we need to restrict the DNA. All I want is the gene for insulin if I want to make insulin, right, in the lab. So reverse transcriptase, what do you think, just by its name, what might it do? Do you remember what transcription is? Transcribing is rewriting. What am I rewriting? The genetic code. So if I'm not making DNA, what am I making?
RNA.
Okay, so transcription is going from DNA to RNA. So what is reverse transcription?
RNA to DNA.
Right, yes, which is why the flu and HIV constantly changing, because they use reverse transcription. So they're starting with RNA and then they have to go to DNA. So if we think about what we know from each of these words, it also helps us eliminate those choices. Okay, there's more on Khan Academy if you want a refresher course on that whole replication process. I sped through it, tried to hit the pieces that I felt were newer to us. Okay, so next week we're going to hit transcription translation. Over the weekend, you'll do those notes. What questions do you have?